Okay, so our first objective here in terms of the physiology is to describe how the urinary secretion of solutes and water influences the volume and composition of plasma and identify other processes that affect plasma volume and composition. So really talking about how do we fine tune that filtrate, which is our plasma being filtered, and how do we create urine? So this slide goes over some of the functions of the urinary system. We spent quite a bit of time talking about that. So I'm going to skip right past this slide and we're going to go to this slide. So here we can see that demarcation that I was talking about earlier. We've got this outer paler staining region called the cortex. This is where we have our renal corpuscle, which is our glomerulus plus our Bowman's capsule. We've got the medulla, which is deeper staining, and this is where we have the loop of Henle, right? This has a descending limb and an ascending limb, and this is intimately involved with the vasa recta. Now, what I want to point out here is, can we see how the capillaries are really tightly wrapped around the entire nephron? This is that reabsorption and secretion that I was talking about earlier. So when we filter our blood out into this Bowman's space, we don't just leave it at that, right? The remaining tubular structures, starting with the proximal convoluted tubule, we can see how this part of the nephron is really twisty, really bendy, and this is the proximal convoluted tubule, but it's wrapped around by these capillaries. And these capillaries are how we reabsorb or reclaim important nutrients that we need to remain in the body, that we don't want to lose in the urine. In the loop of Henle, the vasa recta is intimately wrapped around this region. This is how we modify our water balance. So again, this is how we can reabsorb water, reclaim water. And then the distal convoluted tubule is also intimately connected with these capillaries. And so substances can move from the tubular structures across to the capillary or vice versa. So that's that reabsorption or secretion that I was talking about. Okay. Okay. Now, when we look at the exchange, the renal exchange, there are three main processes that are involved here: the glomerular filtration, reabsorption, and secretion. So the glomerular filtration is the movement of plasma across the glomerulus and into the Bowman's capsule. And that is how we filter our blood into the urinary space, or the Bowman's space, as I called it earlier. Now, I'll talk about the membrane that this has to go across, but basically, this is a selective membrane. It's selectively permeable. So some particles are too large, like red blood cells. They do not get through into the filtrate. Proteins are too large to get through into the filtrate, and so they remain in the capillaries themselves, in the glomerulus. And then the other liquid parts, which is basically plasma and a bunch of other important nutrients like glucose, amino acids, ions, they can freely filter out into the urinary space. And so this is why we have to reabsorb them, because they can get through these spaces. And so the second process is called reabsorption. So reabsorption is movement of important things that we want to reclaim back into the body from the Bowman's space, from the tubular structures, back into the capillaries that are wrapped so tightly around these tubular structures. And then the third process is secretion. And these are things that we don't want in the body, things that probably did not get filtered out, but they're still remaining in the blood, right? Things like excess bicarb, excess hydrogen ions. We want to secrete them. Potassium is also secreted. We want to get rid of some of those things, and we want to move them back into the tubular structures, and we do so via secretion. Now, after all of this fine-tuning of reabsorption versus secretion, we then have our ultimate filtrate, which at this point we call urine, and that can be excreted from the body. So what we're going to go on to speak about today and for the rest of our meeting regarding the renal system is what happens along each of those segments of the nephron. There's a unique set of processes that happen in the proximal convoluted tubule. There's a unique set of things that happen in the loop of Henle. There's a unique set of processes that happen in the distal convoluted tubule. And so we're going to look at step by step what takes place at each of those areas of the nephron and how that ultimately leads to the creation of what we know to be urine.